So let's start. Thank you for joining this session. I'm Soumyadeep from IIT Roorkee, Department of Biosciences Bioengineering. I'm one of the PMRF TAs, NPTEL-PMRF TAs for Tissue Engineering course by Professor Vignesh from IIT Madras. These sessions are from NPTEL-PMRF TA-ship scheme. So this is week 4 and hope you have gone through the contents and the exams are already released and week 5 content is already released. Right? So this week what we have learned about the various surface characterization techniques as well as mechanical characterization and biological characterization techniques. The surface characterization we have learned about the contact angle measurements, AFM, XPSM, etc. In mechanical characterization we have studied UTM, the universal tensile machine, as well as geometer, etc. Right? And biological characterization we have studied in vitro, in vivo assays and hemocompatibility tests, cytotoxicity tests, batch assays, etc. Right? So these are the stuff that we have learned this week. Now let's start with a very simple straightforward question. So the question says that which of the following is not a biological characterization method for a biomaterial? Options are protein absorption study, biocompatibility study, thermogravimetric analysis and bacterial addition study. Just a simple straightforward one. What do you think? What will be the option? Which of the following is not a biological characterization method? Right, I am getting the responses. Yeah, so it is thermogravimetric analysis, right? Right. See, others are for biological characterization, protein absorption study, also blood compatibility, so hemocompatibility assays, bacterial addition study, right? Yeah, thermogravimetric analysis, TGA. Right. So the basic principle of TGA you have studied. There has been discussed like with varying temperature, what are the weight changes of the sample that is happening from very high temperature, from very low to high temperature. This TGA analysis is done just to see what are the how the material behaves. Okay, so it is not a biological property; it is more of a polymer's physical property measurement analysis technique. Okay. So the next question goes XPS, that is X-ray photoelectron spectroscopy, helps in understanding the following except atomic composition, oxidation state, quantification of elements, and weightability of the material. X-ray photoelectron spectroscopy helps in understanding the following except atomic composition, oxidation state, quantification of elements, and weightability of the material. What will be the correct answer? Okay. Anyone else want to try? Okay. Okay. So the correct option is weightability of the material, right? Yes, yes. Weightability of the material. See, XPS analysis, as we have learned, is for elemental characterization. Its oxidation state, its quantity. If we are getting the whole map, then what are the elements that are present, or if we are getting individual peak, that is that is a short form. That is individual peak we can analyze. That is for the oxidation state measurement. So it is for the atomic composition or oxidation state or quantification of elements. This kind of analysis can be done with this XPS. Weightability of the chemical, what we have done, we have used goniometers, right, for contact angle measurement. So that is done through goniometers and contact angle analysis. Weightability of the material, not through XPS. See, that is except option is there. Very carefully you have to study the questions first. That is what is it asking. It is asking what are the options that is except, that is not the correct answer, right? So very carefully. Okay. All right. Next one. Next question. It's more of a practical one. You want to measure the porosity of your sample. What technique among the following can you follow for the purpose? Option goes BET method, NMR, mercury porosimetry, and ACM. Among these, which are the techniques that you want to use for measuring the porosity of your sample? What do you think? What would be the answer? It is a multiple choice one. Try to recollect from the porosity measurement. All right, I'm getting options. Right. Okay, most of you are uh, replying as C, mercury porosimetry. Other techniques that was mentioned in the lecture was this ACM also, right? ACM also was mentioned. So from the lectures, we can say that C and D can be the options, and there are other methods also that I want to discuss here. That's why the correct option is all four: BET method, NMR, mercury porosimetry, and ACM. All four can be used for porosity measurement. ACM, you can visually measure the porosity using their either inbuilt software of the machine, or you can use ImageJ software. It's a Java-based software, ImageJ from NIH. So you can use that software also, or these three techniques: BET method, NMR, mercury porosimetry, and same. These four techniques can be used for porosity measurement. How and why this BET and NMR we can use, we'll be discussing. Okay. So the BET surface area analysis is a widely used porosity measurement technique. Now, theory for this measurement technique has been there from 1938, and it is developed by Stephen Bronner, Emmett, and Taylor. Okay, so this theory based on the based on the gas adsorption. Mind it, it is AD adsorption, not the absorption. Okay, gas absorption phenomena. So the classical techniques of this kind of porosity measurement, which is based on the Langmuir theory. Langmuir theory states that there is a monolayer adsorption of gas molecule on the surface, and it is and this these adsorbents are on the solid surface. Okay, and on a fixed temperature, we can determine the amount of gas that is adsorbed on the surface using the formula. This formula we can use, and we can get the adsorbent quantity, and from there we can get the porosity analyzed. But it has some problems associated with it. That is, these are the limitations, because we cannot make sure that there is only one layer of gas that is adsorbed. So there can be multiple layer of gas molecules that can be adsorbed on the surface, and this different adsorbed layer does not necessarily interact. Okay, and this adsorption theory can be applied for each layer also. So we cannot particularly say that this amount of gas that has been adsorbed, and this is the porosity. We cannot say from this Langmuir theory. So what BET theory states is that this surface is cool to the liquid nitrogen temperature, extremely cold temperature, and nitrogen gas is pumped into the chamber. Okay, and using the following considerations, following equations, they are minimizing the factor that multiple layer of gas molecule that can be adsorbed, and minimization of that factor and using these formulas gave us nearly perfect measurement of porosity of the material. Okay, so BET method therefore precedes from the other classical techniques by considering this this following considerations, that is this steps. Okay, so and this formula, using these formulas, they are considering that the monolayer adsorption and how they are calculating the monolayer adsorption, and the porosity measurement, so that the multi-layer adsorption phenomena of the other classical theories can be minimized, and we can get a nearly perfect porosity measurement. Okay, so this is one classical BET analyzer instrument. It is in IIT Indore, I guess. So this BT instrument, you can see this is for cooling down the sample. There is a liquid nitrogen chamber, so the sample is dipped inside, and using this nitrogen and helium gas, the gas is pumped, and then measurements are done. Okay, so this is the, and you can see in the right panel, instrument diagram, the how, what is the components of the instrument and how the instruments are laid out. Okay, so BET surface analysis technique is also there for measuring the porosity, and I have mentioned NMR also. Though NMR we have seen that for it is for chemical analysis only, how the bonds are there, what are the elements present in the sample. NMR analysis can also be used for porosity measurement. How? And it is not new. They have done it in 1980, 88. Yeah, 1988. They have got this thing out. So what they have, this thing is based on the phenomena of transverse proton relaxation behavior. So think about it this way. Say we have a solvent, water. So water, that OH bond, it behaves differently when it is in the bulk, that is, it's free. All the OH bonds are free, or it is bound to the surface. Now, how this property is different? So this relaxation phenomena, this relaxation behavior changes when it is binding to the surface. Okay. So if we can use that property, and if we can take these relaxation parameters and use it using the NMR instrument, then we can measure the monolayer adsorption of the molecule that is in the sample. Using that factor, it's very crude way of understanding this. So using this property of transverse relaxation, uh, this kind of dipolar relaxation parameter, they have used it, and it's a classical electron, a classical NMR instrument, they have used it, a low magnetizing NMR, and they have combined the software and this instrument to get the porosity measured also using NMR. Okay, and this is a tabletop device, like we can keep it on the table, not like the classical NMR instrument that we have used that requires a whole room. So this kind of instrument and the software can be used like a pharma. It has been used extensively in the pharma industry, where this regular measurement of particle, what are the characters of the particles, or what are the material characters, what are in the surface. This kind of measurement can be done using this kind of instrument. Okay, this is a commercial instrument. You can go through this link also to study the paper. That is, this is the original paper from Graves and Smith et al. So they have given this study in 1988, and based on this study, this instrument has been patented, and this has been out in the market for the surface analysis. Okay, so all the four techniques, BET, NMR, mercury porosimetry, as well as SAME, can be used for porosity measurement. That's why I said all the four options are correct. So whenever in the questions you see among these options, you can choose. Okay, right. So let's go to the next question. Which of the following microscopy has higher resolution? Scanning electron microscopy, confocal laser scanning microscopy, fluorescence microscopy. Which among the following has higher resolution? We have studied it in the week. We know from the resolution, what is resolution? Ability to change, to define the 2 points, right? We have studied about +2 studies resolution. Which among the following microscopy has a higher resolution? Right, I'm getting answers. Yeah, anyone wants to try? Right, scanning electron microscopy. Yes. Why? Because electron is used instead of light. In the other two options, confocal as well as fluorescence microscopy, both uses light for the microscopy. But here, scanning electron microscopy, we have electron beam instead of light. So electron has lower wavelength. So we have visible light based, this laser microscopy, visible or UV light based, whatever it is 200 to 800 nanometer. But electron, you can go way beyond, right? So the resolution is also higher, right? So what are the electrons that are used for imaging? What kind of electron in ACM is used for imaging? What is detected? Secondary electrons. Okay, secondary electrons are detected. We have a specific detector in the ACM instrument, the AC detector, secondary electron detector, that detects the secondary electron. So the electron beam bombards in the surface, and then the secondary electrons that are released, that are detected using the secondary electron detectors, and we get the image, right? And for what is the difference for SAME and TEM? Scanning electron microscopy and transmission electron microscopy. Just crudely, you can say what would be the difference? ACM and TEM. You can say what kind of measurements that we can do using ACM and TEM. Just try. ACM we get the surface properties, while TEM we get the inner properties of the material. So transmission electron microscopy is the electron beam is transmitted through the sample. So what are the sample properties in at the core? From ACM we cannot get the core information of the material. If we have a particle or if we have a slab of material, we only get the surface information, surface morphology of the sample using ACM. Because yeah, what is the basic difference of ACM and TM? What kind of measurements, the measurement oriented differences in the ACM and TEM? ACM we can only get the surface information, right? Because the electron beam is there, and then what are the secondary electrons that is getting out from the gold coating or the metallic coating that is there on the sample. The secondary electrons released from that sample layer is only being detected by the secondary electron detectors. The electron beam is not getting inside the sample itself. While in case of TEM, the electron beam is transmitted, that is going inside, and then whatever the electron that is left, and it is the detector is below the sample, right? Here, the detector you can see in the below. But in case of CM, the detectors are in the sidewise. So the electron beams are coming in, there is the sample, and the detectors are here in the sidewise. Okay. So the secondary electrons that is being released is detected by using this AC detectors. That is the surface properties we are getting, not the inside core properties. While in the TEM, the detector is below. So whatever the sample is getting treated with the electron beam passes a very high voltage, much higher voltage than ACM is used in case of TEM. TEM, there is like a 200 kV or 300 kV electron beam is used. So the voltage is high, very high. So the electron power is also high. So it goes through the sample, and we can see the inner properties of the sample. While in ACM, we get the surface properties. Clear? That's why porosity measurement and everything we can do with ACM, because we can see the surface morphology and somewhat to the depth of the material, somewhat, not like totally in the whatever it is there in the depth. But using the TEM, we can see the whatever the inner properties, what is the material property, if there is any graphs or any kind of composite formed with any nanoparticle graph or anything that is there inside the sample, we can see using TEM, right? Yeah. Should I proceed next? Right. Okay. Next question goes: Universal Testing Machine, that is UTM, is used to determine the following: except tensile strength, compression strength, bending, and surface roughness. UTM is used to determine the following except tensile strength, compression strength, bending, and surface roughness. Right. Right. Anyone else? Right. This surface roughness, right? So what is the technique that we use to determine to measure the surface roughness property? We have studied about it in this week. Try to remember. For surface roughness measurement, no. we can get the surface morphology, but not the surface roughness. For surface roughness, the quantitative determination of surface roughness, we have a better techniques. Better technique that is called atomic force microscopy, right? You remember there are three methods: contact, non-contact, tapping methods. And using that, you can get the surface topography, and you can get a map of the surface. And there you can also get a surface roughness. Surface roughness value is average surface roughness value. You can get quantitatively. So you can cite in the paper that we have the surface roughness. Yes, atomic force microscopy (AFM) we can use for surface roughness determination. Okay, right. So moving forward. So let's think about in a practical situation. So you have given a task of checking if the scaffold you have fabricated can help cellular migration. What test would you perform for the same? You have been given a task of checking if the scaffold you have fabricated can help cellular migration. What test would you perform for the same? Hemocompatibility, mutagenicity, scratch assay, or XTT assay? What would be your answer? Think about it. You have a material. You want to see your cells, whatever the cells you have, that can migrate. So, what would be the test you would be performing? Options are hemocompatibility, mutagenicity, scratch assay, and XTT assay. Okay, okay. Anyone else? Right, try. Just try. Whatever the option that is coming to you. In the last lecture, that is week 4 last lecture. Yes, yes. Last lecture there was a biological characterization, right? So there it was discussed that cellular migration study can be done using the scratch assay. Right? Scratch assay can give you the analysis of cellular migration. You can remember the cells that are grown in a monolayer, there is a scratch was introduced, and then after the introduction of the compound that might have the effect of the cellular migration induction. So that kind of material has been introduced, and it has been shown that the scratch is filling up with the cells that are dividing and migrating to the scratch, and it is being done. That is the scratch assay, right? Hemocompatibility, mutagenicity, you can remember the AIMS assay, where there was a mutagenic bacterial strain with a particular amino acid deficiency. If there is a mutagenic element that is there, if your material is mutagenic, and if you grow the bacteria, if you try to grow the bacteria, the mutagenicity will introduce the amino acid into their system and it will grow. Otherwise, it will not grow. Yeah, that is the test. That is the mutagenicity test we are talking about. Now the XCT assay is also the cytotoxicity measurement assay. You remember, right? The MTT assay, XTT assay. So these kind of assays are there for cytotoxicity measurement. But here the question was cellular migration studies. What can be done? Scratch assay was there. Okay. So try to think about this phenomenon, whatever you are studying there. So try to think about it. You are doing it. You are doing the experiments. Why you are doing the experiment? Why you are doing the hemocompatibility study, virginity study, right? The practical, like if you think that you have synthesized a material, what are the characterization techniques you would be performing depending upon the type of material that you have synthesized, right? So you can remember the things that you have studied this week, that is a week 4, better in this way. Okay. All right. Moving on to the next question. Right. A person visits your lab with a white foam material. It's also a practical question. A person visits your lab with a white foam material and asks for suggestions regarding determining its chemical composition. What would be your suggestion? What techniques he should perform? He or she should perform. SAD, STM, FTIR, and NMR. Among these techniques, what would be the technique that you would suggest to that person? try to think what are the techniques for chemical composition determination. Okay, it's a multiple choice. You can give more than one answers. What would be the possible answers of this question? Right. So both FTR and NMR. Right. These two techniques are chemical composition determination. SAED and STM we have studied and be discussing it also. So these are the material property determination techniques. But the chemical composition you can get using FTR and NMR. And what are the other technique for the chemical composition determination? What we can use for surface chemical composition determination? What is the other technique? We have just studied. TLC, yeah, you can do. And we have just studied about it. Right. XPS, SIMS, yeah, we can do it. Right. So XPS we have studied. XPS is also used for chemical composition determination for the surface. Okay. So these are the techniques. Right. So I was talking about SAED. What is SAED? It is selected area electron diffraction. This is the analysis we do when you are going for a TEM analysis of your sample. Right. So this gives the electron diffraction pattern to you can study your crystallinity nature. So if your sample is monocrystalline or it is polycrystalline, it is a single crystal material or there are different all the various the kinds of crystals are there or various layers of crystals are there in your sample. You can go through the analysis using this SAED pattern analysis. Okay. So this SAED is done through TEM and the lattice measurements can be performed using this SAED. So what it is basically is, for example, see it has three kinds of spots of spot pattern. From left we can start. It is a no diffraction pattern. That is a it is just a white bright spot you can see. Okay, and the second spot is you can get the regular arrangement of spots in the whole field. And the third one is you can see the ring of arrangement of spots. It is a ring-like arrangement. So what is a ring? It is basically the ring-like arrangement of spots only. So why this kind of different spots are being created? So if we think in this way, in the middle there is a single crystal material that is you are studying. Okay. So you have the electron beam going through the sample. Now, if your material that you are studying is a single crystal material, then what is a single crystal? It is a regular arrangement of atoms in a lattice. Okay, so this regular arrangement will also result in the electron diffractions regularity. So if the electron is being applied on the material, the electrons will be diffracted. If the arrangement of the atoms is regular, then the diffraction pattern will also be regular. So this diffracted electron beams will also meet in a regular point, and these points are these spots. So various diffraction electron beams are then coming into a spot, and depending upon the crystal arrangement of the atoms, this regularity of the spots we can find. So this regular arrangement of spots can only be found for single crystal material where there is only one arrangement of atoms inside your sample. Now if we have the sample with multiple layers of crystal or various other crystal types, in that case these spots are not going to be just this kind of regular one. So there are one regular arrangement, then for the next layer that is the atoms are oriented in a different way, the spot is going to be slightly rotated, and for various layers this rotation will lead to the circular kind of arrangement of the spots. So it will not be a like a regularly spaced like spot kind of arrangement for the single that we can find in the single crystal material analysis. For the polycrystalline material, we can find this kind of a ring-like arrangement. And why we are not getting any kind of diffraction pattern for the amorphous material? This is amorphous material. So it is not, there is no particular atomic arrangement for the amorphous material, right? There is no crystallinity in this interface. So the atoms are not regularly arranged. So their diffraction pattern is also not regularly arranged. So whatever the electron beam is bombarding on them, the diffracted electron beam is also getting into everywhere. So there is no electron beam diffraction. One more thing is that whenever we are taking this SAED pattern, there is always this sharp object that you can see. All the papers you see, there is always a sharp or this kind of object. This is not necessarily have to be this kind of triangle-like shape. It can be of any shape, but there needs to be one object that is there. What is the purpose of this? So the purpose is it saves the detector from the direct application of electron beam. So detector might get damaged because of this high power electron beam that is coming through. So it needs to be saved from the direct application. So it it only needs to detect the diffracted pattern. It doesn't need to detect all the electron that is going through the sample, right? So to save that, we need to give a breaker. So this will just act like a breaker. So the electron beam directly doesn't go to the uh, detector and harm the detector instrument. Okay. So this is the SAED measurement. So SAED pattern gives us the idea about the crystallinity and the crystal arrangement of the atoms in the sample. Right. And the next technique is scanning tunneling microscopy. We have studied briefly in the lecture. So I just want to add some details. This STM technique uh, has given the Nobel Prize in 1984, and it's been developed by Rover and Binning. So they have come up with an idea that there is a quantum mechanical phenomena called the tunneling of electrons that happens when there is two conducting materials very close together. So if you can think about it this way, whenever there is a conducting material and cloud of electron hover around the surface of the material, but this material, this electron that is hovering around is cannot go through much distance because of the barrier property. The electron doesn't have that much of a power to go through like everywhere. So what it can do when there is another conducting element very near, that is less than 100 nanometer distance, there can be a current flow. That is, the electron can jump to the other conducting material of opposite polarity. Right? So this phenomena is called tunneling current, and based upon this tunneling current, and so when there you can see the instrument layout. So it is controlled using a circuit where they're trying to put the voltage of the circuit same. So when there is a tunneling current behavior is happening, if there is, for example, say there is a rough undulation and the probe that is going through, if it comes to the that rough peak, and so the distance between the sample and the probe decreases to the value where there can be a tunneling current increase. So what the circuit would try to do, it will try to decrease its own voltage so that the it can be kept in the same balance. So this detection of voltage difference or current difference, we can measure the properties of the sample or the whatever the information, that is surface information of the sample, we can get through this STM, that is scanning tunneling microscope. Okay, so what kind of materials that are used for the tip? That is tungsten, platinum iridium, and gold. This kind of conductive material is used as a tip, and you can see from this image, this kind of patterns we can get from a sample. So this is the atomic level information that individual atoms can be mapped using this STM instrument, because we can get the information whatever the atomic arrangement or whatever the atoms are present and what kind of atoms are present. We can also get the information, not a direct measurement of the like particular measurement of the kind of atoms, but the atomic arrangement we can also get through this STM measurement. So you can see this regular arrangement of atoms, as well as there are certain this kind of bulky So these are the defects in the sample. So our crystal, there might be some defects atoms that are not, that should not be present in that lattice, but it is there. So, or there is no atoms in that site, or there might be some atoms in the lattice, in the surface of the lattice, where that should not be there. That is the air atoms. So this kind of detection can also be done using this HTM. Challenge is there because it is based upon this voltage and current measurement. So it needs to be a conductive material for the measurement to be done, and there might be certain vibrational interference that if the sample is vibrating very slightly also, the detection pattern will change. And there, if the sample is having any kind of contamination, very small dust particle or anything in the sample surface, the detection will be different. Okay, the modification of this technique is the AFM. So AFM uses this probe and the laser deflection. That is, when the probe is there using either contact mode or tapping mode or non-contact mode. These three modes in AFM we have studied. So, and the probe is being detected with the help of a laser that is being deflected from the probe. So as the probe is getting from and scanning the surface, the laser beam is also detecting the probe's movement. So that's how the AFM is being AFM has come up. So they have given this is the modification of STM only, the AFM, and they have got also a Nobel Prize for this AFM. So yeah, so STM and AFM, these are very important techniques to study about, and this gives the molecular information of the material that you are investigating. Okay, STM, right? Yeah. Okay. So the next question that I want to discuss is. It's a practical one. So you want to study your Mg63 cells behavior upon compound A introduction for 5 days. You want to study your Mg63 cells behavior upon compound A introduction for 5 days. What assay or test would you be performing? Options are Ames test, MTT assay, scratch assay, LMRP assay. What would be the technique? I've given this question for a particular reason. You want to study your Mg63 cells behavior upon compound A introduction for 5 days. What assay or test would you be performing? It is Alamoglu assay, right? So Ames test is not related to this Alamoglu assay. Yeah, I don't know if you're trying to respond as Alamoglu. So yeah, Alamoglu we are using. MTT is a destructive technique. Formalin crystal needs to be resolved using DMSO. Scratch assay is like, it is not for this cellular behavior, like its cytotoxicity measurement. So it is not for that. It is for more like cell migration studies. Okay. So yeah, scratch assay can also be an option, you can say. But yeah, more I wanted to stress upon this Alamar blue assay because this is a non-destructive technique. You can wash the media with the after the measurement and then reintroduce a fresh media, and then you can go with the culture because it is not going inside the cell. It is from the media property only. So if it is analyzing the media changes in the media, this Alamar blue technique can also be used for as a non-destructive alternative. You can check the cellular behavior for a longer period of time. Right. And why this Mg60 cells and compound A introduction is, it is just to distract. So many times the questionnaire may think about introducing this kind of Mg60. It is a osteo, it is a cell, a bone related cell. It is a cancer cell. So this kind of cells name and compound, the various compound names can also be there just to distract. The try to focus on the question itself, what they are want to study and what they are focusing on studying. Not the what kind of cells they are studying or what kind of compound they are introducing. Not always it is important. Yeah, it might be important sometimes, but this kind of questions, that direct question is not asked in general. Okay, so try to focus on the kind of question, that kind of problem they are giving, and what kind of answer they are expecting, not the what are the cell line or what kind of compound that is being used in the question. Okay, right. All right. So before going into the before going to asking what are the questions that you have now, I wanted to address some the discussion forum question. I always encourage you to ask various questions in the discussion forum, and you have responded. I am really thankful. And there are other questions also in the discussion forum that has already been answered. I thought I can discuss these questions uh, here in the live session also. So there was one question that was asked, that is, it goes like, in case of solvent casting, after placing in silicon mold, we leach the salt by immersing it in water. If the polymer is hydrophilic, will it not swell? If so, how do we get a dry scaffold? We are betting that the polymer that we have used is hydrophilic and that it swell, because after it swells and it is cross-linked, and when we are leaching, we are just dissolving the salt that we have used in case of salt leaching method, so that the salt portion only goes away, but the because of the cross-linking, the polymer will stay stable. And after we have done that, we will go for lyophilization, and then we will get a dry scaffold. Okay, so think about this way: you have a hydrophilic polymer, not the hydrophobic one. It won't work with the hydrophobic one, right? So you have a hydrophilic polymer, you have taken it, and you have mixed it with the salt crystals, and then you have casted it in a silicon mold. Then you have introduced the cross-linker. It has done the cross-linking, then, and it is already in a bath of it is already in a solution bath. Then you can take that sample and then places in another solution bath, that is maybe the water or PVS, whatever that is, and you can leach out all the salt that is there in the structure right now, right after the cross linking is done. And then for the leaching, you take it to the PVS or water. You have a beaker or maybe a petri dish, and you have placed it and put water in it or PVS in it, so that salt gets leached. Water would be preferable because of osmotic reasons. All right, so you have done that, and then you take the sample and freeze dry it using lyophilization, and you get the dry scaffold. Okay, it's clear. Next, the question goes like, if we are to do swelling study, initially the weight could be X, and after swelling, the weight of the swollen scaffold could be Y. Right. Just after this question, I will be repeating. Just give me 2 minutes. So, so after swelling, it gets to Y. Okay. When it reaches equilibrium, there may not be any intake of water. Thus, at some point, the scaffold may start to diffuse the water out, leading the weight of less than the previous time points. Right. In such cases, how do we analyze the swelling study? So when we are doing the swelling study, what we are doing is we are taking the dried hydrogen, putting it in a water or the solvent or whatever the buffer solution that we are using. We are putting it in there, and then we are thinking that we are getting the hydrogel being swollen. As soon as it gets to the equilibrium point, we take the hydrogel out, tap dry it using a tissue paper, and we measure the weight or the dimension of the hydrogel. We do not let it dry for long, and this equilibrium, to reach to the equilibrium, the water is getting into the hydrogel matrix, and it is stable. It doesn't leach the water out that much easily. So we are tap drying it to get rid of the surface water that is there. We just go for immediate measurement of the weight and dimension of the scaffold, that is hydrogel, and then putting that back into the buffer. So we are not letting the water get out of the matrix. But yeah, there might be, if you leave it for a long time in a dry environment where the humidity is less, the water might get out and you will get a different data. There is a possibility. So try to do it whenever we are doing this swelling studies. Do it as much early as possible. So you let it get to the equilibrium, take it out, tap dry, and then do the measurement. Right. For the solvent casting technique is what is done. But in case, even if it is a hydrophilic polymer, there is no issues because we are cross-linking it. There is a cross-linking region either physically or chemically. There are various cross-linking techniques. So when we are placing the hydrophilic polymer with the cross-linker and salt solution along with it, so the cross-linker will do its job. It will be cross-linking the polymer. And then when we are taking the slab out and putting it in a water or buffer, the salt will leach out, and even if it is hydrophilic, that is not a problem because we have the stable cross-linking of the polymer chain. So it will not wash away. All right. So, and to get the dry one, so the when we have the salt leached out, we'll just lyophilize it. That is a freeze drying method to get the dry scaffold. Right? Clear? Okay. All right. So the next question is, what is sintering and what is rheology? The sintering is the process where the material is subjected to a very high temperature. Okay, so the temperature that creates a possibility of the materials of various kind to diffuse. So what it does is there is a neck like thing that is created while there is a sintering is going on. This necking is there, and this will ultimately lead to the fusion of the material. So there is two three things I want to quickly say. It is depending upon the diffusion property of the material, right? That is the diffusion behavior. It depends, and it can be either surface diffusion or bulk diffusion. Okay, so when there is a rise in temperature, the material might fuse depending upon the diffusion parameter at the elevated temperature. It can be either surface diffusion, it can be depending upon the defect flux, one thing. Secondly, it can be a grain boundary diffusion of the material, or it can be a bulk transport of the material. These three things can be there at the elevated temperature. So how the sintering is done? This sintering is done using boat, a ceramic boat, and you place the sample and you get that material into a very high temperature furnace, where the temperature can reach up to 1000 degree or 1000 or 1500 degrees, and you do this sintering. And the temperature range depends upon the type of material. If we have a metallic material of high temperatures resistance, then in that case the temperature can be higher than 1000 to 1500. Okay, that high temperature can also, it can be burned. So you can think of sintering as a as kind of a burning process. For our hydrogel property, or in case of a ceramic sintering, it can be like a 300 to 500 range. The temperature can vary, and it is for our purpose. It is just like to burn out the scaffold, whatever the polymer scaffold we have used for giving it the structure. So it can be for our purposes, ceramic material purposes. It can be like a burning out process. Okay, and to fuse the ceramic material to a single phase, right? So that is sintering. And you have also asked about the rheology. In our hydrogel perspective, there are two parameters that we most of the time hear. That is the storage modulus and loss modulus. So what are those? I want to discuss. So for rheological measurements of a particular hydrogel, there is one method, that is one model, that is the two plate model. What is two plate model? The bottom plate, if you think about it, there is two parallel plates. Okay, and you have put the sample upon the bottom plate, and it is sandwiched between the top and bottom plates. The down plate is fixed, and you can move the top plate, and you can give the top plate a rotational motion. So it can be like a sinusoidal motion of the top plate. It can go to the particular angle, then come back, and so this kind of motion, rotational motion, it can be given to the sample. Now this top plate model can be used for rheological measurement, and depending upon this sinusoidal motion and the property of the material. So if you think about it this way, that the top plate is moving in a particular sine wave, right? But the material is not conducting that movement efficiently to the bottom plate. So bottom plate has a load cell or a detector, you can say. So it is not detecting at the same time the top plate is moving. So there is a delay. That delay is called the phase shift, and it is also that is denoted by delta. This phase shift value is just like it is a shift in the sine waves because of the, and depending upon the property of the material. If it is a really elastic material like a metal or something, then that kind of material will transfer this movement really efficiently, and there is no lag between this, or the delta is zero almost, right? But in case of a fluid or a viscoelastic material, there is a viscous range and the elastic range. Okay, we'll discuss. So in case of that kind of material, this transfer of this motion, that is the sinusoidal motion of this top plate, it is not that much efficiently transferred to the bottom plate. So there would be a phase shift, that is called the delta. And from this information and the measurement of the stress, shear stress amplitude and strain amplitude, that is also can be measured using this top two plate model. So and this we get the complex shear modulus, that is the G star, and it is a vector parameter, right? So we have studied vector in the plus 2. So it is a vector parameter, and it can be split into x and y direction components. So this x component, belongs to the elastic contribution of the parameter, right? Whatever the material's elastic property, that is a solid, that is there, that property is denoted by this x component contribution, while the y component contribution denotes the loss modulus or the viscous component. So what happens in case of hydrogen? We have the polymer matrix and the fluid, that is water inside the material. So this polymer property will be denoted, that is a polymers only the structure, that is a solid state phenomena, that we can give through the x-axis contribution, while the y-axis contribution will be it's the viscous component, that is the water that is there. So why it is so this x component is called the storage modulus, that is the hindrance of this. So while changing the this rotation, this sine movement, there would be a hindrance to get to to that bottom plate, the sine wave movement. There would be a hindrance because of the solid component, right? So this is why it is called the storage modulus, right? And the loss modulus, because of the water content, there would also be a hindrance property because of the water content. So that property is called the loss modulus, and it can be denoted by G double prime. So G prime and G double prime, these two parameters, that is the y-axis contribution and x-axis contribution, this we most efficiently measure in case of hydrogen characterization. And the tan of this value, delta, this tan delta, this is called the loss factor, that is G double prime by G prime. This gives us the idea about the property of the material. If the delta value is closer to the x-axis, that is lower, less than 45 degrees, this is the material is mostly solid. Okay, and if the delta value is greater than 45 degrees, the viscous component is more, so that is mostly fluid in nature. Okay, so viscoelastic polymers shows this kind of properties, and we measured this G prime and G double prime, the storage modulus and loss modulus. If you read the hydrogel papers, you can see this measurements using the DMA, dynamic mechanical analyzer. They study this G prime and G double prime changes depending upon the polymer's characteristic. Okay. Now, another thing is this stress strain behavior. Is that you have studied about the necking phenomena in case of uniaxial tensile stress strain, right? So the metal and the polymeric material behaves very differently. Okay, so when the metal is stretched, then necking is created just very little before the fracture point. So there is not much movement of the material after this necking starts. But in case of polymers, you can see the necking starts, and all the stretching or the elongation of the material as the strain is being induced. So this happens in the neck region only. Okay. So what happens is, in case of polymers or viscoelastic materials, this neck region, there is a molecular arrangement of the polymer of the monomeric unit of the polymer. So as the strain is being introduced, there is continuous molecular arrangement, and this fracture point is reached at a very long after the necking is there. So this different kind of properties of this inertial stretching behavior depending upon the necking introduction, after what happens after the necking introduced in the metals and what happens after the necking is introduced in polymeric or viscoelastic material is different. Okay, because of the behavior how metal and polymer, this both two are the poles of the materials kind of thing. So how differently they behave. Okay, so this a book, Callister's book is there on the material science and engineering. Two chapters I will suggest you read. Chapter 6 for metals characterization and chapter 15. 10th edition probably. You can go through this link and you can study these two chapters to uh, study more about this polymer characterization and what are the properties, how the stretching inertial strain behavior, how the mechanical characterization is performed. That has been mentioned very beautifully and very elaborately, and how the mechanical characterization is done. Okay, and so Uh, with this, I want to conclude my part of the discussion. I would wish to ask you if you have any questions. Please do ask from the week's content, other than the discussion forum points that has been already answered. If you have any other questions, so I hope you are going through the chapter contents and whatever and the lecture slides that are given, and you are doing well in the exams, right? Okay. If you have any questions, right? So with this note. Uh, I hope I have conveyed the message that I wanted to discuss in this week's discussion. Uh, and I would again wish to encourage you, please write to the discussion forum with all your questions, and hope we are being able to answer your questions. If you still have doubts, you can write and you can directly ask here in the live sessions also, and we would love to answer them. And ask as many questions. Don't hesitate to ask and study the course materials very uh, attentively. Okay, and thank you for joining, and hope you join next session also. Okay, right. Thank you for joining. You can leave the session.